Hello TGT aspirants, welcome back to a new video of Sunanda's tutorial. As I have promised, I will come with a video of describing your chapter sexual reproduction in higher plants. So it is your last unit of botany syllabus. So let's start. At first, what is sexual reproduction? So the reproduction which involves two parents and hence generate a new combination leading to variation. So I have already posted in my community section about the asexual reproduction or vegetative propagation modes of vegetative propagation. So please go to my community section and watch those post about the vegetative reproduction in plants. So sexual reproduction, in case of sexual reproduction, it involves two gametes, one male gamete, one female gamete. They combine to form a new gene combination which leads to the variation. Flower primarily serves as the organ of sexual reproduction. The ultimate aim of sexual reproduction is to produce seeds for the next generation. Each individual flower is born at the tip of a specialized stalk called pedicel. The apex of pedicel, where floral organs are attached, is usually more or less enlarged, called thalamus or receptacle. So suppose it is a flower, it is the stalk called pedicel. It is the enlarged portion called thalamus. All other parts are attached to this thalamus. The outer hole is called calyx, then inner to it corolla hole is present, which is the colorful part of the flower. So it is the calyx. This colorful part is the corolla. It is the thalamus. It is the pedicel. Then inner to it stamen or male reproductive part is present, then female reproductive part. Two outer hole of the flower, that is corolla and calyx, is called as the accessory hole, and the inner two hole is known as the essential hole because they take part in the reproduction, sexual reproduction. So, center of receptacle is occupied by gynoecium or pistil, which is the female reproductive part. Actually the calyx is the outermost hole and individual unit called sepal. Then corolla individual unit of corolla is known as petal. Then androecium or male sex organ, individual part is called stamen. Stamen consists of one filament with two anthers, bilobed anther at its terminal. Then gynoecium is the female reproductive part or pistil. The individual unit of pistil is called carpel. It also have three parts, one enlarged portion called ovary, then the long stalk called style, then it is the stigma. It have three parts. All process of sexual reproduction except pollen grain formation occur in the ovary. Next, events of sexual reproduction. At first, microsporogenesis, formation of pollen grain, then microgametogenesis, two male gametes are formed, then pollination, transfer of male gametes to the female part, then megasporogenesis, formation of megaspore, then megagametogenesis, formation of embryo sac. A typical, then the details about the male reproductive part and its development. At first, a typical anther is dithecous. Dithecous means filament is attached to a bilobed anther, which is dithecous. Each of these sac consists of two spore sacs and called as dithecous. Then tetrasporangiate. Tetrasporangiate means actually the structure of a mature anther is like this. It have four spore sacs, so tetrasporangiate. Then structure of a matured anther. So it is the structure of the matured anther. It consists of the outer wall is called the epidermis, then inner to it endothecium. It consists of elongated cells with fibrous thickening, but some endothecium lack fibrous thickening, and they help in the anther dehiscence, that means release of pollen grain outside the environment after maturation. Then some cell without thickening called stromium. Then inner to this endothecium 3 to 4 middy layers are present. Then inner to it tapetum, which supplies the nutrition to the growing pollen grain. These are pollen grain. Okay. Then the development of anther. So under the development, at first young anther is there. Soon it becomes 4-lobed. That young anther is actually consist of some parenchymatous cell surrounded by epidermis. That's all. But when it matures, slowly the hypodermal cells begin to act as archesporal initial. Each archesporal initial divide into primary parietal cell and primary sporogenous cell. Parietal cell divide periclinally. Periclinally means horizontally. It divides horizontally in 180 degree to form endothecium, middle layer and tapetum. And sporogenous cell undergoes mitosis or equational division to form microspore mother cell. Microspore mother cells undergoes meiosis to form 4 haploid microspores at the end. Okay. So at first parenchymatous cell surrounded by epidermis is there. Then soon it becomes 4-lobed, and then some hypodermal cell act as archesporal initial. It divides to form parietal cell and sporogenous cell. Parietal cell forms endothecium, middle layer and tapetum. Epidermis is already there, and sporogenous cell undergoes mitosis to form microspore mother cell. Microspore mother cell undergoes meiosis to form 4 haploid pollen grains. So these are the arrangement of pollen grains. Generally they are present in tetrad. Tetrad means 4 cells are arranged together. They may be they may show tetrahedral arrangement, they may show isobilateral arrangement, they may show dicoset arrangement. So it varies. Then microgametogenesis. The pollen grain, actually the released pollen grain have this structure. They have outer exine which have sporopollenin. Then actually the sporopollenin is very tough, very thick and very hard. They are biologically inert. None of the enzyme can degrade that sporopollenin. So they preserve in the environment for long year, for million of years. But at some points this pollen grain, the outer wall, have some pore. They lack sporopollenin and called as germ pore. Then inner to exine intine layer is there, and intine layer consists of pectocellulosic. It consists of pectin and cellulose. Then inner to intine nucleus is present. So during the process of microgametogenesis, that nucleus divides into vegetative nucleus and generative nucleus. Generally pollen grains shed at this 2-celled stage into the environment. Then the female part, female reproductive system and their development. Pistil or gynoecium is the female reproductive part. It consists of an enlarged ovary, a long style and stigma, and sometimes it consists of one carpel called monocarpellary. Some flower consist of more than one carpel called multicarpellary. Sometimes these carpels are free from each other called apocarpous. Sometimes they are fused with each other called syncarpous. Inside the ovary many cavities are present called locules. Suppose this ovary have 2 locules. Inside that locules ovules are present. The portion of ovary to which ovules are attached called placenta, and that arrangement of ovules inside that ovary is called placentation. Then ovule, mature ovule consists of a stalk and a body. One end of funicle is attached to the placenta. The point of attachment called hilum. Funicle ends extend up to base of the ovule and a ridge is thus formed called raphe. Body of ovule have 2 ends, one micropylar end, one chalazal end. Ovule is covered by 1 or 2 envelopes called integuments. That integument have an opening called micropyle. Integuments enclose a large parenchymatous tissue called nucellus. In the center of nucleus, female gametophyte or embryo sac is present. So ovule or megasporangium is a structure like this. Many layers are arranged like this, called as integument. Inside integuments, nucellus is there. Inside the nucellus, embryo sac is there. It have two end. This ovule have two end. One is micropylar end, one is chalazal end. Then inside it, eight nucleus and 7 cells are present. We will discuss what are those cells and nucleus later on. So now we will discuss how this structure, this ovule is developed. At first, earlier in young stage, subepidermal cell of nucellus act as archesporal initial, and one specialized cell act as megaspore mother cell. It divide to form primary parietal cell and primary sporogenous cell. Primary sporogenous cell develop to form megaspore mother cell. Megaspore mother cell. Megaspore mother cell undergoes meiosis to form 4 megaspores. Then 3 of these megaspore towards the micropylar end degenerate, and one lowermost megaspore present at the chalazal end remains functional and forms an embryo sac. Functional megaspore always forms the embryo sac, and the above process that I have discussed, subepidermal cell act as one subepidermal cell act as archesporal initial. Then it divide to form megaspore mother cell. Then megaspore mother cell divide to form primary parietal cell and primary sporogenous cell. And 3 of the megaspore degenerate. One act as functional megaspore. It forms embryo sac. So till the formation of a megaspore is called as megasporogenesis. After 3 megaspore degenerated, then one functional megaspore act as active member, and from that active megaspore whole embryo sac is formed in the process of megagametogenesis. Where this type of development is occur called as monosporic embryo sac, where from one megaspore embryo sac, whole embryo sac is formed. Okay. So this one functional megaspore undergoes 3 successive mitotic division, from 1 to 2, then 2 to 4, then 4 to 8. So finally 8 nucleate embryo sac is formed. 6 surrounded by cell wall, 3 here and 3 here is surrounded by cell wall, 3 at micropylar and 3 at chalazal end. 2 free nuclei move to center called as polar nuclei. Okay, so this is the structure of a polygonum type mature embryo sac. Then after that pollination occur, transfer of pollen grain to the female reproductive part of the plant or stigma. It can be of 2 types. One is cell pollination or autogamy. It can be jetenogamy, and it may be cross pollination. So see, in cell pollination, the pollen grain from the same plant will be transferred to another flower, to another flower or same flower but born on the same plant. But in case of cross pollination, the pollen grain transferred to one plant to another plant, flower of another plant. In case of cell pollination, it can the same flower, the male part, from the male part of that same flower, pollen grain is transferred to the stigma of that same flower, called as autogamy. Sometimes it is transferred to the flower born on the same plant but different flower, called as jetenogamy. Then the cross pollination or allogamy or genogamy, where transfer of pollen grain occur from stigma to the from the male reproductive part to the stigma of a flower born on a different plant. So plant adapt some contrivances or some adaptation. Plant shows some adaptations to carry out the self-pollination. These are homogamy. In some plants, stamen and carpel mature at the same time. So pollen grain can easily pollinate the stigma of same flower. Cleistogamy is another adaptation where flowers do not open even after maturation. And cleistogamy is shown by the underground flower of Commelina vegalensis. Some plants also show adaptation for cross pollination. One is dicliny or unisexuality. Actually, some flowers are unisexual in nature, so cross pollination is obligatory there. Secondly, some show self sterility or self incompatibility. Pollen grains from the same flower do not have any effect on same flower stigma. Then dichogamy, stamens and carpel in some plants mature at different types. Sometimes they show protandry where anther mature earlier. Sometimes they show protogyny where carpel or female reproductive part mature earlier. Then horcogamy. Some plant species have a physical barrier between anther and style. Then heterostyly, length of style different in individuals of same species. So these are contrivances or adaptations to self pollination for cross pollination. Then fertilization after pollination, fusion of male and female gamete occur called the fertilization process. This process was first discovered by Strasburger, and this fusion results in a diploid zygote. After pollination, the pollen grain received by the stigma. Pollen grain produce the pollen tube after germination. Pollen tube carry the male gamete to egg cell, and that process is called siphonogamy. Because pollen grains of higher plants are not motile, so they cannot move by itself. So pollen tube carry that male gametes to the egg cell, the process of siphonogamy. Then step 1, germination of pollen grain on stigma and growth of pollen tube. Stigma first recognize the pollen, and if it is compatible, only then it germinates, otherwise it is rejected. Pollen tube emerges from germ pore. The solid style have specialized tissue called transmitting tissue, along which the pollen tube grows towards the embryo sac, and that pollen tube contains vegetative nucleus and 2 male gametes. Then entry of pollen tube into the ovule. It can be porogamy when pollen tube enters through micropyle, which occur in most of the angiosperm. Sometimes it shows challazogamy, where entry of pollen tube occur through chalaza length, for example in Casuarina. Sometimes it shows mesogamy, where pollen tube enters through punicle or integument, for example in case of Pistacia and Cucurbita. Entry of pollen tube into embryo sac. After entering into the ovule, it will enter inside the embryo sac. The pollen tube enters into the embryo sac only through the micropylar end. In the embryo sac, pollen tube passes between synergids and egg cell. And sometimes it enters into one synergids through filiform apparatus. Actually, filiform apparatus guides that pollen tube to the egg cell. Then true fertilization or syngamy occur. One male gamete fuse with female gamete to form zygote. Another male gamete fuse with the polar nuclei to form primary endosperm nucleus. It divide to form endosperm, which is triploid in nature. This double fertilization was first demonstrated by Novaschin in 1898 in Fritillaria and Lilium. Zygote then develop into embryo, and primary endosperm nucleus develop to form a mature endosperm. Then development of dicot embryo. How dicot embryo is developed from the zygote. At first, zygote divides transversely or periclinally or in 180 degree to form a basal cell and apical cell. Basal cell divides transversely, apical cell divides longitudinally, so it forms a T-shaped proembryo. Then it have a basal cell and 2 apical cell. Basal cell actually forms your hypophysis, then radicle, then root cap, all that things which are present in root section. And apical cell divide to form globular embryo, then heart-shaped embryo, then mature embryo have cotyledon and plumule, and from plumule the whole shoot system is developed. So from radicle, root is developed, from plumule, shoot is developed. Then development of monocot embryo. Zygote divide to form basal cell and apical cell. Basal cell have 2 zones, C1 and M. It's just number. It's just a letter to signify 2 cells, 2 region actually. Then C I region divide to form N, which develop to form a part of root cap. N dash, which is the outer cell, forms the suspensor. M region divide to form the remaining part of root cap and radicle. Then apical cell divide to form a quadrant embryo. Then quadrant embryo have peripheral cell and inner cell. That inner group divide to form scutellum and coleoptile. Scutellum is the cotyledon. Actually monocot have one cotyledon. At first one cotyledon is developed, and coleoptile is the second leaf. And the part of embryonal axis below scutellum is called radicle, enclosed by coleorhiza. So it is basically the brief and point of very short points of development of zygote to embryo from the zygote. Then endosperm which is developed from the primary endosperm nucleus. It can be of three types: nuclear endosperm, cellular endosperm, and halobial endosperm. Nuclear endosperm, where primary endosperm nucleus divide freely into large number of nuclei without any immediate cellular formation. Liquid syncytium of coconut, which we drink, is actually the nucellar endosperm. Then cellular endosperm, division of primary endosperm nucleus is immediately followed by wall formation. At first, two equal sized chamber, chalazal and micropylar end, is formed, which is followed by regular cellular formation. Then halobial endosperm, primary endosperm nucleus divide and form two chamber. One is micropylar chamber, one is chalazal chamber. Pre-nuclear division occur in both chamber. Divisions are not followed by wall formation. So these are three types of endosperms. Apart from that, just remember these types of endosperm. One is ruminate endosperm that shows irregular and uneven surface contours on the surface, is called ruminate endosperm. Mosaic endosperm, patches of different colors of endosperm tissues occur. Then what is genia? Just remember that effect of pollen grain on endosperm is called genia, which is observed by maize. Then metagenia, effect of pollen grain on tissue outside the endosperm, shown in case of date palm. Then albuminous seed possess large amount of endosperm, as in wheat, maize, castor. Then ex-albuminous seed that lack endosperm, for example pea. So it is all about your reproduction in higher plants. In my community section, I have already posted about the vegetative mode of reproduction. Please watch that community post, and I hope you will do your best in your tomorrow's exam. All the best to all of you, and please like my videos, share as much as possible with other aspirants, and please subscribe to my channel Sunanda's tutorial. Thank you and all the best for tomorrow.